This is 5 things you should know about genetics and Dravet, and this is a very parent-focused session. So it should be a primer if you want to go back to the CME session. The doctor's version of their genetics PowerPoint is kind of after this one. So hopefully you'll have some idea what they're talking about if you choose to go back. And this is everything I'm going to say, really. The 5 things you should know about Dravet are that SCN1A codes for the sodium ion channel. We'll talk more about that. Mutations change the code. Mutations are common in Dravet syndrome, and not just SCN1A mutations, but other mutations too. They can be new to the child, or they can be inherited, and you have the power to decipher a lab report. You can do it, and we're going to walk through a couple of them and show you how to read your child's results. And then no presentation is complete without a shameless picture of your own kid. So that is my mutant. He has a mutation, and those are the X-Men who also have mutations. Unfortunately, they got a little bit luckier with theirs than Aiden did. But all kids with Dravet syndrome are superpowers because of their mutations, just like the X-Men. So just a little bit of stepping back from genetics just for a second to talk about seizures and what they are, because those will play into how the genetics come into Dravet syndrome. We all learned in high school biology about the cell. We all made the models, right, with the pasta and the organelles. We labeled that mitochondria and that endoplasmic reticulum. But what they didn't teach us in high school is that cells actually come in all different shapes and sizes. And the neurons that you see in the small box up there are actually, those are single cells. And they are totally differently shaped than those big circles that you saw in biology. And one of the things that they have on them that's important to our kids are sodium ion channels. They're like little pores in the membrane of those cells, and they let charges into and out of the cells. And the way that the brain talks to the body is by sending electrical impulses. And the way that they do that is they change what's called the gradient, meaning the difference in charge between the outside of the cell and the inside of the cell. And the way that they change that gradient is by sending ions into or out of the cell. And the ion channels are what the sodium ions go through. Remember, sodium is that it's part of table salt. It's an ion. It's charged. It's one of the ones used in neurotransmission. When those ion channels don't work properly, abnormal electrical impulses result, and seizures can be a symptom of that. So the ion channels that we saw in that last picture in the membrane are that protein that you see with the green. It looks kind of like the cross-section of a donut a little bit. The ions that are actually going into and out of the cell are those little blue circles. So that's the goal. That's where we're going to get to, and where we're going to start is way back at the DNA. So also remember from high school biology that DNA is in the nucleus of our cells. That's like the brain of our cells. And the human body has over 3 million base pairs in their DNA, meaning if you remember that double helix, it looks like a ladder. Each step on the ladder is a base pair or a nucleotide, depending on whether you're looking at 2 of them or 1 of them. And the body, every single cell in your body has over 3 million of them. Billion, thank you. Over 3 billion of them. And so when we are looking at SCN1A, we are looking at a tiny, tiny, tiny fraction of the DNA in our cell's nucleus. And when the cell goes to read that DNA, it reads it in just a section, and it knows where to start because there's a start codon, and it knows where to stop because there's a stop codon. So there are little signals on the DNA that say, hey, start reading me here if you want to make an ion channel. And then the cell reads it, reads it, reads it. And then when it gets to the end, there's a stop codon that says, hey, stop reading me here, you're done. If you keep going, you're going to read something else. And when the cell is reading that, it is translating that DNA from DNA or RNA is one step of the problem, into amino acids. And amino acids are kind of like the building blocks of life for humans. All of your DNA codes for building proteins. That's its only job. So your cell can make many, many, many things that it needs just by reading the DNA, translating it into a string of amino acids, which then fold to become a protein. That middle step, that amino acid chain, I like to think of it as building a necklace, stringing beads on a necklace, and stringing them in a very, very specific order, and picking from a group of 20 different beads. So there are 20 different amino acids that the body uses to make proteins. So you have 20 choices for the beads, And those 20 beads all have different chemical properties. Some of them are attracted to each other. Some of them repel each other. Some of them are attracted to each other at the top and not the bottom. So when you string this necklace and you have all these different beads with different chemical properties, then when you go to put it in your drawer, you kind of clump it up, it's going to make a 3D shape. And that 3D shape is going to be determined by how those beads interacted with each other, whether they were attracted, whether they repelled. And so if you have the wrong beads on that chain, you are going to get the wrong interactions, and the final result is that that protein is not going to be the right shape. So the two kind of take-homes from this is that DNA determines the sequence of the beads, and the beads determine the shape, and if it's not shaped right, it won't work right. So stepping back to that first process where we saw the DNA or RNA, the DNA making the chain of amino acids. We have 3 billion base pairs in our DNA, but your body reads them in groups of 3, and they're called codons. So every 3 base pairs, and the base pairs are represented by letters, and they can be 4 letters. It's usually A, C, G, or T in DNA. If you're talking about RNA, that T is switched for a U. So it's A, C, G, and U, or T, that you'll see in these slides. They're read in groups of 3. The groups of 3 are called codons, and every single codon specifies a very specific amino acid. So it calls for a very specific bead. So in this instance, that first codon, GCU at the top, calls for the amino acid alanine, abbreviated ALA. And there are some crossovers where you can have GCU, for example, and GGU, and that those both might code for alanine. So there's a little bit of play in there, but the take home is that every 3 letters codes for a specific amino acid or a stop codon. So codon 7 there is UAG, and that means stop. And those specific letters mean stop. And a stop codon often starts with a U in the RNA sequence. Not always, but sometimes. So when you make this amino acid chain, you start at a specific place, you put things in the specific order, and you stop at a specific place. Any deviation from that can be detrimental to the final protein or the ion channel. One more thing on this. The SCN1A has over 100,000 base pairs, or rungs on the ladder, the little steps, in its unedited form. But the body will actually edit a lot of that out. We don't know why it's there. It may have been there because we needed it evolutionarily years ago. We don't know what a lot of that is doing. And so the body will take sections of it and it will splice it out, or it'll cut sections out, and it will splice those sections together. And each of those sections is called an exon. SCN1A has 26 exons. And so in its final edited form, there are 6,000 base pairs, which I'll do a little bit of math here. What's 6,000 divided by 3? Okay, so there are 2,000 amino acids in the final sodium ion channel. So we have 6,000 base pairs. Divide that by 3, because we read it in groups of 3. There are 2,000 amino acids in the final protein. You with me so far? Okay. So the second thing that everyone needs to know about genetics and Dravet is that mutations change the code. There are several different classes of mutations. The easiest one to explain is the missense mutation. A missense mutation is just a simple substitution of one letter or one base pair for another at a specific spot. So in this case, that second amino acid, threonine, was changed to proline because that first A in the second codon was changed to a C. That one change changes one amino acid, and remember there are 2,000 amino acids in SCN1A. So you would think one bead on the necklace out of 2,000 probably shouldn't be that big of a deal. I know if I were doing quality control, I probably wouldn't notice that. But our bodies are so sensitive that in a lot of our kids, that one change out of 2,000 things can lead to Dravet syndrome. At the same time, that one change doesn't always lead to anything. Sometimes you can have a missense mutation that you don't even notice because it's in a healthy individual. The healthy individual was never tested because there was no reason to test them. Or you can have a missense mutation in somebody with generalized epilepsy with febrile seizures, or just febrile seizures themselves. It can cause a wide variety of symptoms, but it can be anywhere from mild to severe. So missense mutations are easy to explain. Their result is not so easy to explain. The second type of mutation that you've probably heard about are called nonsense mutations or termination mutations or truncation mutations. They all are versions of the same word. In this case, the mutation actually happens somewhere along the line, and it changes the codon from coding for an amino acid to coding for a stop codon. And that's premature because the body wasn't supposed to stop reading the gene there. It was supposed to keep stringing beads on the necklace. But unfortunately, that one codon that it changed to codes for the word stop, and so it stops reading it. And the result of that is a truncated protein, meaning your necklace, instead of being 2,000 beads long, might be only 1,000 beads long, depending on where that is. It might be 5 beads long, or it might be 1,999 beads long. In general, it doesn't seem to matter where that truncation mutation happens. It often leads to a non-functional protein. And though there are cases of nonsense mutations causing mild symptoms, more often than not, they lead to Dravet syndrome, which is the severe end of the spectrum. I don't know if you remember from Dr. Locke's or Dr. Miller last night was talking about the spectrum of SCN1A, and there was mild, and then there was severe, and Dravet is at the severe end. But then we all have Dravet syndrome, or our kids have Dravet syndrome, so trying to decide whether it's mild Dravet or severe Dravet is, all bets are off. No one can really do that based on mutations yet. All we can say is that it's probably Dravet, or there's a chance it might not be. But in terms of Dravet, no one can decide the severity yet. So still with the idea that mutations change the code, you can have what's called a frame shift mutation. So remember that the body is reading the DNA in groups of 3 called codons. If instead of just substituting one letter for another one, instead you mess up and you actually put in an extra letter, then that's going to change the reading frame for every single codon after that mutation. And so what happens is that you might get a protein that's just as long as it should have been, but it's all wrong. It's all the wrong beads from the place that frame shift mutation happens on is completely wrong. And that can happen with either an insertion of a base pair or a deletion of a base pair. Or it can be an insertion of 5 base pairs. It can be a deletion of 5 base pairs. It can be any number. It doesn't really matter. The result is the same, that everything from that point on is nonsense. Now, back when we learned biology, these terms were a little bit confusing because nonsense mutations 20 years ago, when I learned biology, included frame shift and premature stop codons because it basically said that it was nonsense, that this was not a readable gene and it wasn't making a functional protein. That terminology has changed, and so now when you read lab reports or you're reading scientific publications, they will specifically say a nonsense mutation. They're talking about a termination mutation. They're talking about a premature truncation. I hope that's not any of our kids. And they'll usually specify that frame shift is different than nonsense, whereas before we kind of lumped them all into the same group. So there are a couple other types of mutations. One is called a splice site mutation. Remember how we talked about the body edits SCN1A down from over 100,000 base pairs? It edits it down to 6,000. Well, where'd those other 94,000 go? So anywhere at those splice sites where it went to cut that DNA, you can have a mutation there, and it may mean nothing, or it may mean Dravet syndrome, or it may mean not Dravet syndrome. Again, this is one of those ones where you just really can't tell. So the lab reports, when they specify a splice site mutation, they will usually say, we don't know what this is going to cause. And again, like the frame shift mutations that we were talking about, you can have very large deletions and insertions. You can have a deletion of the entire SCN1A gene, usually causes Dravet syndrome or something on that spectrum. You can have deletion of not just SCN1A, but SCN2A and all the other genes near it. So there can be any length of deletion or insertion. So now that we understand, hopefully, a little bit what the mutations are, what does that mean for our kids? we already know that mutations are common in Dravet syndrome, and not just SCN1A mutations. There are 70 to 80% of our kids, or 7 to 8 out of 10 kids, will have an SCN1A mutation, but the other 2 to 3 don't. Those other 2 to 3 may have a mutation in PCDH19, which is more often found in girls, and results in a phenotype, which is what you see. Phenotype, by the way, is like the clinical what you see. Genotype is your actual DNA. Phenotype, when you hear the people in the CME sessions talking about phenotype, they're saying how it presents, what it looks like. The girls with PCDH19 present very similar to Dravet, but there are a couple differences, and so they're often misdiagnosed with Dravet, and then when they go back and test them, they may have a PCDH19 mutation. There are some that have both mutations. There are some that are unlucky. And then the other 2 to 3 kids out of 10 will either have no mutation that we found, or they might have a mutation in another gene that for some reason presents just like Dravet, which is okay. Remember, Dravet is a clinical diagnosis. So not having an SCN1A mutation does not mean you don't have Dravet. It just means either we haven't found the mutation because our tests aren't sensitive yet, or you have a different mutation. So then looking at the mutations that we know of that have been reported, that first graph on the left is from a database of mutations published in the literature. So this is my compilation of it. So this is not published data, but the data that created these graphs is published. Of the known SCN1A mutations, and these are mutations that were caught in people that had symptoms. So remember, we're not really testing healthy people for the most part unless they're parents, and nobody's reporting that data. So these mutations that are out there are published in the literature. These people had some sort of symptoms. Of the known SCN1A mutations, about 75% of them had Dravet syndrome, and about 24 to 25% of them did not have Dravet. They had Geff's plus, or they had febrile seizures, or something milder. So that's pretty telling that just because you have an SCN1A mutation does not mean you have Dravet. Dravet is a clinical diagnosis, but 75% of the people that we found with SCN1A mutations do, so there's a good correlation there. So then taking the blue part of that pie graph, the Dravet SMAI diagnosis, those mutations, and then separating those out is what creates that second graph. And in the mutations that we know of that have caused Dravet, in cases published in the literature, about 40% of them are missense. So missense was that simple substitution where it just changed one amino acid, it just changed one bead on the protein. About 40% of our kids have that, and there's really no way to tell whether they're going to be severe Dravet or whether they're going to be mild Dravet or what they're going to be. We can't find a correlation yet. 20% of them will have a nonsense mutation or termination mutation, and 20% of them have a frame shift, and about 10% have a splice site mutation, and 7% have some larger portion of the gene either deleted or inserted. Some of the literature that you'll see out there will say 40% missense, 40% nonsense. That's that problem with the terminology that we were talking about before, where they used to lump the nonsense and the frame shift together. Now they kind of separate them out. So you might see 40-40 rather than 40-20-20 sometimes. So now that we know where our kids stand in terms of everybody else in the room and what they have, how did this happen? I actually can't answer that, but I can say when it happened, usually. So mutations can be new or inherited. About 90 to 95% of our kids' mutations are de novo, meaning the lab asked for the parents to be tested, and the parents sent their blood in, and the blood tests came back negative. So they said, okay, this is a new mutation in the kid. The parents don't have it. Great. So we all think we're all set. We can go have more kids, and there should be no problem because it just happened in the child. What that can actually mean is that it's new to the child, but it may have been present in the egg or the sperm. And so we don't actually know if the parent doesn't have it somewhere in their body. They may have it in their egg or sperm. We don't know. All we know is that the mutation showed up for the first time in the child. So that de novo, that 90 to 95% de novo is a little bit misleading sometimes because even mutations in the germline, which is the egg and sperm, can appear to be de novo. They can also be inherited. There are cases of the exact same mutation being inherited from a parent, and the parent has mild symptoms, or the parent has no symptoms, or this is a completely shock, and the parent says, I had no idea I had problems. And it's inherited in an autosomal dominant fashion, which means that, remember, okay, high school biology again, we all have 2 copies of each gene. One comes from the egg, one comes from the sperm. So we all have 2 copies. Autosomal dominant means that even if you have one bad copy, you're going to have symptoms. And that is the pattern of inheritance for SCN1A. So all of our kids usually have one healthy one and one mutated one. If it is inherited from the parent, if they find it in the parent's blood, that parent then has a 50% chance of passing it along to every single child they have. So just because you have one that you passed it along to does not mean that chances are your next child is going to be okay. It's 50% every time you conceive. So mutations can be inherited, and there's a phenomenon called reduced penetrance. So a lot of people say, well, why does my child have Dravet syndrome, but I have the mutation and I'm fine? And that's actually not as uncommon as it seems. It's not as odd of a phenomenon. This is known to happen in biology. For some reason, we don't actually know why, but it's not just SCN1A, it's other disorders as well. Because of reduced penetrance, it can have different phenotypes. The same mutation can have different phenotypes. About 3 to 10% of Dravet syndrome mutations are inherited directly from the parent, so we know, like we've tested the parent and they have it. A new type of inheritance that's kind of gaining more attention in the past couple of years is called mosaicism. 10 years ago, when we were testing for mutations, we'd test the baby or the child and find a mutation, then we'd test the parents and not find the mutation, say you're good to go, no problem. Well, genetic tests have gotten a lot cheaper, they've gotten a lot more widely used, and they've gotten a lot more sensitive, and it turns out now we're finding that in some of those parents that we said did not have the mutation, more sensitive testing is revealing that they do actually have the mutation in just a small part of their blood. And so it wasn't showing up on the first test because it was a very small percentage of the blood that was mutated. And the way that can happen is by mosaicism. So if you have a healthy egg and a healthy sperm, and at fertilization they come together, then they come together as one cell. That one cell divides and becomes 2 cells. Those 2 cells divide and become 4 cells, and on and on and on. If you have a mutation that happens spontaneously sometime shortly after conception, only the daughter cells from that mutated one are going to be affected. So if the mutation happens, let's say in this case it's happening at the 4 cell stage, only the ones passed along from that mutated cell are going to have the mutation. And this is simplified. It doesn't mean that 25% of your blood is going to be mutated. It really depends on what those cells were destined to turn into and where they were going to go in the body. It's not quite as simple as that, but that's the idea. And it is turning out that up to not that recent papers showed that up to 10% of mutations that we thought were de novo may actually be inherited from a mosaic parent. And also in that database where I pulled the mutation frequency from, it also shows that up to 42% of the inherited mutations are from a mosaic parent. So it might be it's more common than we thought. It doesn't mean it's common, but it's more common than we thought. Which brings us to the last thing that you need to know about Dravet syndrome and genetics, and that is that you can decipher a lab report. They are not written in Greek. You can do this, and we'll walk you through it. So this is my son's lab report. This was back in 2007, so this is pretty old, but it turns out they all kind of look the same still. So the first thing you want to look at is that nucleotide position. So if you can find it up there, I don't know if you can read that, but it's that 4073. So remember, SCN1A has 6,000 places, right? So at 4,073, we're about 2/3 of the way through the gene. And in this place, it was supposed to be a G and mutated into a T. So at that spot, he has a transversion, just one simple letter changed from a G to a T. What that did was it made the 1,358th codon or bead or amino acid code for a different amino acid. It's under that green line. It says it was supposed to be a tryptophan bead. It's a leucine bead instead. This is a simple missense mutation. One letter changed, one amino acid changed, and they will tell us that we don't know what it means. So because a missense mutation can be associated with any kind of phenotype, they'll say, we have no idea what this means. Well, time told us that it means Dravet syndrome in this case. The other thing that they will usually tell you is that there is no other form of this mutation published in the literature. So this lab report didn't say that, but they usually do now. They'll tell you if there's another one. So another example of a lab report would be for a nonsense or termination mutation. You go through the same process. You look for your nucleotide change. So take a minute, see if you can spot it. It's right there. This one is at 4,521. So again, roughly 2/3 of the way through, a little bit more. In this case, a C. It was supposed to be a C. It mutated to a G instead. So it sounds like a missense so far, just one letter changed. But that means that it changed it into a termination codon or a stop codon. And the way you can tell that is that all of the amino acids will have single letter abbreviations like Y or L or A, or you'll see all sorts of different abbreviations. And that Y before the 1507 is the amino acid that it should have been, a tyrosine, and the X is stop. I'm sure that's pretty self-explanatory. It changed it to a stop codon. So this is a nonsense mutation. And it even tells us that. This is a great lab report. This is Ambry Genetics, by the way. They did an awesome job. It tells you it's a nonsense. They also tell you that it is deleterious. Now, that doesn't mean there's a deletion, even though it's kind of a confusing word. It means that it's probably pathogenic. It's probably a disease-causing mutation. So that deleterious is not a deletion. It just means it's bad. And I believe this report also said that there was a published case in the literature, but I don't know if I have that on it. Okay, so another example, if you had a frame shift, your lab report might look something like this. First thing we do is we look for the nucleotide change, right? It's right there. At the 830th place, there should have been a G, but instead it took it out, so it's a single deletion. And remember that taking out that one letter is going to change the reading frame of all of the amino acids, of all the codons afterwards. And so deleting that one letter meant that everything after it is garbage. And so they can say with pretty good certainty, we're going to skip that one for a second, that it's pathogenic because they know that this is making a protein that is probably not functional. So they can say it's going to cause some sort of symptoms. Notice they don't say that it's going to cause Dravet syndrome, but they do say that it's probably going to cause symptoms. So back to that exon intron. When we talked about the body editing out SCN1A down to 6,000 base pairs and doing it in 26 segments, this lab specified that it was on exon 6. It's not really that useful of information, but just if you're curious, that's where it's coming from. It is pathogenic, and this one also says that it has not been reported in individuals with SCN1A-related disorders, but it is expected to cause disease because it is a frameshift mutation. Now, again, the severity of the disease, they can't say. It might be febrile seizures, it might be migraines. They don't know for sure, but it's probably going to cause something. And it probably, if you were being tested, there was a reason you were being tested, they're saying this is probably that reason. The last one that we'll go through is a splice site mutation, and unfortunately this one, the facts did not come through very well, but we're going to go through the same steps. We're going to look for that nucleotide position, and it's right there. It's C, and you can't see that, but it's actually 474. So at the 474th place, remember, out of 6,000, so we're not even a 12th of the way through here. At the 474th place, then you've got something strange there that says -1 G to A. We haven't seen that -1 before because this mutation happened at a place that normally gets edited out. So you look at the 474th position, and then you go back one into one of those sections that actually got edited out, into one of those introns that got edited out, and that's where the mutation took place. So why is that important if it got edited out anyways? It's important because if the body doesn't recognize the right places to edit, then it might not edit out the right things. You might end up with a protein that has information in it or beads on it that weren't supposed to be there. And so splice site mutations, even though they're supposed to be edited out, they are important because if you cut it in the wrong place, you're going to glue it back together in the wrong place. So that's what splice site mutations look like. They'll usually either be minus or plus, meaning it's 1 before this place or it's 5 after this place. This one says it's a splice site variant. It says it's pathogenic, likely to cause disease. The only way they knew this, by the way, that it was pathogenic, was because it has been reported in the published literature. So splice site mutations in general, you can't really say what's going to happen with them because it's very hard to know whether it's going to be an important one or a non-important one. So the only way that they knew that this was likely pathogenic was because they had seen a case of it before. So with that, we have -1 minute. Are there any questions?